The AMP-PCP is an ATP analog, and ALF is the aluminum fluoride species, where it looks like the iron protein is plugged into the MoFe protein. So this would be the plugged-in conformation. And then there are these other two conformations where the metal centers are farther apart by about 5 angstroms that probably would slow down electron transfer by several orders of magnitude, that you would consider the unplugged states or off states. And this turns out is coupled to the conformation of the iron protein, in that there's one set of conformations that are consistent with this ALF type complex, where there's a fairly short distance that should be favorable for electron transfer. But then when the ATP is hydrolyzed to the ADP, it stabilizes this more open conformation with a significantly greater, about 5 or whatever, 6 angstroms greater distance. And so we think that one thing that ATP is doing is basically creating a, we'd say, a bistable switch. So it's really a two-state system. It's either on or off. And that nitrogenase is a pretty slow enzyme. It's working in the presence of protons that can be reduced to hydrogen. And so a challenge nitrogenase faces is trying to minimize short circuiting, so electrons going to reduce protons to hydrogen. And so you have a system where you're able to plug in these two proteins for a length of time that should be sufficient for electron transfer to take place. But then instead of allowing the electrons to return, since eventually they'd reach equilibrium, the complex dissociates. And so this essentially, I think, you sort of open the door, you throw an electron, you close the door before the electron can come back out. That's what ATP is doing. Okay, well, this is just sort of a hypothetical animation. I guess animations are hypothetical, but anyway, trying to show this sort of interaction. Here's the iron protein. You can see in different states of the nucleotide here that here you have the close distance, and then when the phosphate groups leave, this is associated with a more distant sort of the unplugging. There we go. As near as we can tell, the most significant, whatever one means by significant, conformational changes are in the iron protein. And while we see small changes in the MoFe protein, it does sort of seem like it's a rock with very few changes taking place so far. Okay, so we've been, really started this work to understand how nitrogenase works. And so we'd like to have some idea of how dinitrogen comes in and interacts with its metal center and gets reduced to products. So I was at some meeting, and people working on ribosome were giving these impressive talks with these really elaborate movies. And so I was feeling a little depressed because I didn't have a movie. But the nice thing about working with a diatomic is it's easy to make a movie. And so I decided so I came up with an animation, also hypothetical, that describe sort of my understanding of how nitrogenase works. And so this will be in real time. Okay, so there you go. It takes about a second. It takes about a second, and something happens there in the middle. So, okay, so eventually, hopefully not in another 20 years, we'd like to understand. And so there are a number of things, really, I'd say the most important part is to understand the mechanism of substrate reduction. And we're working through here trying to see if this center may change in more highly reduced states. What is this ligand in the middle? Does it matter what it is? Where does substrate bind? What does the P cluster do? And then one last thing I would just end on is these are really elaborate metalloclusters which have really been challenging for synthetic chemists to produce. And so far, there are no really good models for the iron molybdenum cofactor that have been made synthetically. And we probably shouldn't feel too bad about that, because the biosynthetic pathway by which these clusters are made is quite complicated. It involves a number of intermediates and precursors and so on. And we've been working with Marcus Ribby at UC Irvine, and recently determined the structure of a protein NIF-EN that has a similar fold. So it's a homolog to the MoFe protein. But in this case, it's being used, this protein, NIF-EN, plays a role in the biosynthesis of the iron molybdenum cofactor, in that this little blob here is a precursor presumably an iron and sulfur only containing form that eventually will become this when it grows up. And so we've been looking based on these similarities. We've seen that NIF-EN is similar to NIF-DK, which is the MoFe protein. These are the names of the genes. And we have in what would the counterpart of the alpha subunit cases where some complex apparently can dock to the surface. It then goes inside a cavity, and probably at this point something happens in terms of either electron transfer chemistry or other ligands binding. This cluster then gets transferred to a form of the MoFe protein lacking the cofactor to make the mature protein. And so far we've been able to determine structures for 3 of these 6 states, and hopefully we'll figure out ways to trap these clusters in these other states as well. So anyway, to conclude, I've been working on this project for some time. I can really appreciate the role of mentorship and teachers and colleagues in carrying out scientific research and education. This started, Jim Howard was a sabbatical visitor in Lipscomb's lab when I was a graduate student, and he convinced me that nitrogenase was an interesting system. And so it sounded, good to me. So I did a postdoc in Jim's lab, and the time at UCLA and so on, I'd say, was pretty slow going. Nowadays, probably one would not, at least I wouldn't have survived this first 11 years, at least in the academic sense. But I've been really fortunate to have a series of really wonderful students and postdocs working in this project, and so it's a pleasure to acknowledge their contribution, and I'd like to thank you again for this opportunity to present the Goldstein lecture. Thank you.